The last lab, fruit quality and inspection and generation. So the summary of it is focused on understanding and interpreting how a ResNet 50 computer vision model makes decisions rather than training a model from scratch. So we set up a fruit quality inspection model. We work with a fruit datasets containing images of apples, mangoes, tomatoes, and each fruit had two conditions. So healthy or fresh, it will be classified as 0, and rotten is classified as 1. So we load an already fine-tuned ResNet 50 model that classifies fruit as fresh, rotten, since the model was already trained. So our main goal was to understand what happens inside the model and why it makes a certain predictions. So we made prediction using the ResNet 50. We pass fruit images through the model to classify them as either fresh or rotten. Conceptually, from the fruit images, we use the ResNet 50 and we have the prediction, and the prediction is either fresh or rotten. This gave us the model finals output before we investigate how it reached that decision. Next is we explored the convolutional neural network feature hierarchy. In CNN feature hierarchy, we inspected different layers of ResNet 50, including from the convolutional one, we have the layer 1 index 0 convolution 1, layer 2 index 0 and convolution 1, layer 3 index 0 convolutional 1, and layer 4 index 0 convolution 1. We use forward hooks to capture intermediate feature maps or activations produced by these layers. This helps us understand the CNN's learned features hierarchically. From the early layers, like we have the edges, colors, and textures. In the middle layers, for example, we have gathered the patterns and structures. And from the deep layers, it produces a complex or high-level features. So we observed how the model gradually transformed the original fruit images into more abstract representations. And we visualized the most active feature maps. In the feature map strip, using that function, we analyze the feature maps captured from early layer. We calculate the mean activation of each channel, selected channel with the highest average activation, extracted that feature map, upsampled it into 224 by 224, and normalized the values between 0 and 1. So the process was from the feature maps that has 1 by channel by height and width to mean activation per channel and find the highest activation. We select the feature map, resize to 224 by 224, and normalize, and finally by the visualization. This allows us to see which feature map responded most strongly at each stage of the CNN. And we created the Silency map. In Silency map, we examine these pixels in the input image had the strongest influence on the model's predictions. We cloned it and detached the image tensor. We enabled gradient computation using the request grad, perform the forward pass, select the score for the target class, and perform backpropagation. And we obtained the gradients of the input image, took their absolute values, and combined the RGB channels, normalized the result. And the Silency map helped us answer which pixels have the greatest effect on the predictions. So the brighter areas represented pixels to which the model's predictions was more sensitive. So we generate a simplified class activation map. Using the function simplified CAM, we use the class activation map or CAM to identify larger image regions that influence a particular class prediction. We capture feature maps from the code, model.layer 4, and its index is -1.convolutional 3. We also use the weights for the target class from model.fc.weight, and its index is the variable is class index. We then combine the feature maps and class weights to produce the activation map. So conceptually, we have the final feature maps, and then target class weights, weighted combination, the ReLU, and normalize, upsample, and class activation map heat map. This helps us determine which areas of the fruit model focus on deciding whether it was fresh or rotten. We compare different interpretability techniques. Through the lab, we work with three important interpretability techniques. So, the feature hierarchy, so it is what features the CNN detects at different depths. And saliency map, which individual pixels strongly influence the prediction. And which larger regions contribute a particular crust prediction. A simple way to remember them is the feature maps is what is the network detecting, and saliency map which pixels matter, and class activation map which regions matter. Together, these techniques helps us better understand the internal reasoning of the ResNet 50 model. And then in the last part of the laboratory, we have the stable diffusion section. We loaded stable diffusion. In the optional section, we work with stable diffusion two ways. We loaded the stable diffusion pipeline and moved it to the appropriate device. This introduced us to the idea of using generative AI to create additional training data. We generate synthetic fruit defects. We generate synthetic fruit images using text prompts. For example, we use a prompt describing a mango with a small hole made by a worm in the middle. We also use a negative prompt such as fresh, intact. This demonstrates how this stable diffusion could potentially generate examples of rare fruit defects when world images are limited. So the general process was from text prompt, stable diffusion, denoising, synthetic fruit image. We visualize the denoising process, the denoising movie.